At the end of a clinical trial, when the treatment is complete, there's all this data left over that is rich for learnings. In particular, there's imaging data from each of these patients that describes how the drug worked in this individual. And in the Vol-PACT program, we pulled together these data from multiple different large clinical trials to look back and find new ways of understanding drug effect in patients undergoing treatment.
All too often in oncology clinical trials, the trial may be successful in the early phases of development, but then ultimately in the larger phase 3 trials, there really doesn't turn out to be that great a difference between the experimental arm and the control arm. The goals of Vol-PACT is really to define better imaging biomarkers, to use the existing imaging techniques that we have, but to better quantify the changes on those images and correlate those images and those changes with patients' outcomes. When we analyzed subsets, random subsets of the phase 3 data to create phase 2 trials, it was possible indeed to more accurately predict the results of the phase 3 trial.
I think this is clearly the new horizon, which is zooming in on radiomics, the complicated variables that describe tumor behavior, and behavior on immunotherapy. And our hope is this can lead to accelerated drug development. So we really know which of these treatments are working and which ones aren't. And so we can tell in which patients are they working so that we can be even more precise in how we deliver these exciting new therapeutics.
The FNIH has been really excited about Vol-PACT. We actually consider it one of our flagship projects within the Cancer Steering Committee for the Biomarkers Consortium. It accurately reflects kind of all of the best parts of what the Biomarker Consortium can be. Brings together industry, academic partners, government partners, as well as other associations and nonprofits.
Vol-PACT creates a unique environment where drug sponsors, companies that have this rich data sets feel confident that they can share the data under a foundation of trust and that the data will be analyzed with the utmost scientific rigor and integrity in a regulatory context.
Merck understood the value in contributing to an organization that was established in a pre-competitive space with a mission that really focused on improved efficiencies in clinical trials. When we work with patients, their participation in a clinical trial is exquisitely valuable. So as an industry, we're really obligated to do what we can to optimize every aspect of their participation in a clinical trial.
The next phase of Vol-PACT really involves the prospective analysis of these imaging techniques, or the analysis of the imaging techniques and the metrics that we've already defined. And then to incorporate some of the radiomics features, which our imaging scientists have proven are unequivocally robust and reproducible. We could look at these features that are reflective of a tumor's underlying biology and underlying genetics and see those changes over time.
Vol-PACT started with an ambitious goal, which is to collect all of these imaging data, look again at them with the most advanced technologies, and figure out if we can understand the best ways of capturing how a treatment works in a cancer patient. We want to share our learnings with every stakeholder so they can then take these ideas and run with them.
These models could ultimately help us change the current paradigm and make our clinical trials more efficient and have a better link to clinical outcomes for our patients.
When we put that together, we will then have really a true success for the industry and for patients undergoing cancer care.